Hello students, today's topic is cryopreservation. In this session, we will discuss about the different steps involved in cryopreservation for germplasm conservation of plants. Conservation of germplasm can be done by cold storage, low pressure storage, low oxygen storage, and cryopreservation. In cold storage, the germplasm is conserved at temperatures 1 to 9 degrees centigrade. This is a low temperature but non-freezing. The growth of the plant material is slowed down in cold storage. In low pressure storage, partial reducing of atmospheric pressure of the surrounding is done, while in low oxygen storage, reducing the oxygen level but maintaining the atmospheric pressure is done. In cryopreservation, the germplasm is stored at extremely low temperatures of minus 80 degrees centigrade to minus 180 degrees centigrade. Low pressure and low oxygen storage. Here, as it suggests, the germplasm is stored under low atmospheric pressure or low oxygen. In low oxygen storage, the reduced availability of oxygen and reduced production of carbon dioxide reduces the photosynthetic activity. This inhibits the plant tissue growth and dimension. This method helps in increasing the shelf life of many fruits, vegetables, and flowers. Cryopreservation is a method of preserving cells in frozen state. In this method of conservation, the tissues are brought down to zero metabolism or non-dividing state by reducing the temperature in the presence of cryoprotectants. This can be achieved in one of the following ways. First, over solid carbon dioxide, where the temperature is reduced to minus 79 degrees centigrade. Extreme low temperatures in deep freezers, example at minus 80 degrees centigrade. In vapor phase nitrogen, where the temperature is at minus 50 degrees centigrade, and over liquid nitrogen, where the temperature of storage is minus 196 degree centigrade. By means of cryopreservation, the tissues or cells can be stored in inactive state for a very long period of time. The steps involved in cryopreservation of plant material is first development of sterile tissue cultures, then addition of cryoprotectants and pretreatments, freezing followed by storage. For revival of the plant material, it uses thawing followed by reculturing, then measurement of survival and viability and plant regeneration. For development of sterile tissue culture plant, any culture material can be used or explant can be used. Next, addition of cryoprotectants and pretreatments. As in this method, the tissues are stored at a very low temperature, the freezing process or cryogenic temperatures may lead to formation of ice inside the cells. This may lead to irreversible mechanical damage and osmotic shock. This can be prevented by addition of certain chemicals or cryoprotectants like DMSO, glycerine, ethylene, sucrose, mannose. These cryoprotectants also help to protect the tissues during thawing process. During freezing, the water molecules inside the cells form ice crystals and expands. This leads to cell damage. During slow freezing, the water exudes out of the cell and forms ice at the extracellular space. This causes mechanical damage to cell. Besides this, the solutes inside the cell gets concentrated in the remaining water. This too damages the cell. If the rate of cooling is rapid, small ice crystals are formed inside the cell. Cryoprotectants are commonly hydrophilic molecules that inhibit water crystallization and decrease the water chemical potential. A cryoprotectant can be a penetrating or a non-penetrating type. Penetrating cryoprotectants are small in size, example DMSO and glycerol. They penetrate through the cell membrane into the cell and replace the water molecules. They form bonds, hydrogen bonds with the water molecules. This prevents the water molecules from uh, ice formation during cryopreservation. Non-penetrating cryoprotectants are larger molecules like sucrose and polyethylene glycol. They do not enter the cell but dehydrate the cell before freezing. Thus, they inhibit ice growth. They also maintain the osmotic equilibrium of the cell. This would also help during thawing. After treatment with cryoprotectants, the samples are freezed. Various freezing methods are slow freezing, rapid freezing, droplet freezing, and vitrification. Slow freezing method. In this method, the cells or the tissues are cooled at a very slow rate, 0.5 to 5 centimeter degree per minute from 0 to minus 100 degrees centigrade and then transferred to liquid nitrogen. The advantage of this method is only little water exudes out of these cells. This promotes extracellular ice formation rather than intracellular ice formation. The plant cells would be partially dehydrated and can survive better. This type of cryopreservation is usually successful in case of suspension cell cultures. Rapid freezing method. In this method, the vials containing the samples are immersed into liquid nitrogen. This leads to rapid freezing, a decrease in temperature of minus 300 degree to minus 1000 degree centigrade per minute. The process is so fast that small ice crystals are formed within the cells. The growth of this intracellular ice crystals are prevented. This method is useful in cryopreserving shoot tips and somatic embryos. Vitrification. It is an alternative to freezing. It means turn into glass. In this method, the cells are treated with high concentrations of cryoprotectants and then plunged into liquid nitrogen, leading to ultra rapid freezing. In this method, there is no ice formation, as seen in the figure below. The salts and the other molecules that are there in the cells remain undisturbed in their natural locations. Certain vitrification solutions used are DMSO, acetamine, propylene glycol in combination, ethylene glycol, ficol, and sucrose in combination, or ethylene glycol, DMSO, or ethylene glycol, glycerol in combination. This slide lists down the differences between slow freezing and vitrification. In vitrification, if you notice, the cell escapes the ice formation step. The frozen vials are then kept at temperatures ranging from -70 to -196 Storage is ideal in liquid nitrogen at -50 in vapor phase or in liquid phase at -96 Storing cells at low temperatures may lead to loss of viability of these cells. Hence, in cryopreservation, the viability of the germplasm is periodically checked. Proper documentation records are also maintained in germplasm storage. The information should be comprehensive and should have the following particulars: taxonomical classification of the material, history of culture, morphogenic potential, genetic manipulations done, somoclonal variations, culture medium, and growth kinetics. Thawing. This is done to revive the frozen cells and tissues. It is carried out by immersing the frozen samples in ampules into warm water at 37 to 45 centigrade with vigorous shaking. This rapid thawing protects the cell from damaging effects of ice crystal formation. As the thawing occurs, the ice melts and the ampules are transferred into water baths at temperature 20 to 25 centigrade. After thawing, the germplasm samples are washed several times to remove the cryoprotectants and recultured in fresh nutrient media following standard protocols. The cells, when stored at extremely low temperatures, lose their viability. The viability of the cells needs to be checked after thawing. This is generally done by certain staining techniques like staining with Evans blue fluorescent diacetate. The viability of the cells are then calculated. This is followed by plant regeneration. Thank you.